Welcome back to Clinical Hematology Reviews. Today we'll be discussing complete blood count, or CBC, parameters. Automated CBC analysis provides the foundational quantitative framework for laboratory hematology. Modern multi-parameter hematology analyzers rely on a combination of direct current electrical impedance, radiofrequency conductivity, laser light scatter, and spectrophotometry to enumerate and characterize whole blood cellular elements.
Understanding how these parameters are derived is critical for every laboratory scientist. We classify data into directly measured parameters, derived histogram parameters, and calculated indices. Direct measurements include the RBC, WBC, and platelet counts, along with the hemoglobin concentration.
In contrast, the mean cell volume, red cell distribution width, and mean platelet volume are directly derived from pulse height volume histograms. Hematocrit, mean cell hemoglobin, and mean cell hemoglobin concentration are mathematically calculated from those primary values.
Quality assurance demands that we recognize instrument operational limits, validate results against internal quality control checks, and understand specific corrective protocols whenever interferences arise.
Adult reference intervals are established using cohorts of healthy individuals and are reported in both conventional and international system of units, or SI units. For total WBC count, the adult reference interval is 4.5 to 11.0 times 10 to the third per microliter, which translates to 4.5 to 11.0 times 10 to the ninth per liter in SI. The platelet reference range is 150 to 400 times 10 to the third per microliter, equivalent to 150 to 400 times 10 to the ninth per liter.
Red cell parameters demonstrate physiological divergence between adult males and females due to the stimulatory effect of androgens on erythropoietin production. For adult males, the reference range for RBC count is 4.50 to 5.50 times 10 to the sixth per microliter, or times 10 to the 12th per liter in SI. For adult females, the RBC count is 4.00 to 5.00 times 10 to the sixth per microliter.
Hemoglobin for males spans 14.0 to 17.4 grams per deciliter, which converts to 140 to 174 grams per liter in SI units. For females, the hemoglobin reference range is 12.0 to 16.0 grams per deciliter, or 120 to 160 grams per liter in SI units. Hematocrit follows a similar pattern, spanning 42.0 to 52.0% in males, reported in SI as 0.42 to 0.52 liters per liter, and 36.0 to 46.0% in females, or 0.36 to 0.46 liters per liter.
The calculated red cell indices remain uniform across sexes. Mean cell volume, or MCV, ranges from 80.0 to 100.0 femtoliters. Mean cell hemoglobin, or MCH, ranges from 28.0 to 34.0 picograms. Mean cell hemoglobin concentration, or MCHC, spans 32.0 to 36.0 grams per deciliter, or 320 to 360 grams per liter in SI units. Finally, the red cell distribution width coefficient of variation, or RDWCV, normally sits between 12.0 and 14.6%, and mean platelet volume, or MPV, spans 6.8 to 10.2 femtoliters.
When automated parameters breach predefined critical limits, known as panic values, immediate physician notification is mandatory due to imminent clinical risk. A critical low WBC count below 1.0 times 10 to the third per microliter warns of severe neutropenia and high risk of life-threatening infection, while a critical high WBC above 40.0 times 10 to the third per microliter flags a potential leukemic crisis or severe leukemoid reaction.
For hemoglobin, values below 7.0 grams per deciliter signal severe tissue hypoxia, requiring urgent transfusion evaluation, whereas values above 20.0 grams per deciliter indicate severe polycythemia and risk of hyperviscosity syndrome. Platelet critical limits are defined as counts below 50 times 10 to the third per microliter, which predispose to spontaneous hemorrhage, and counts exceeding 1,000 times 10 to the third per microliter, which carry severe thrombotic and microvascular risks.
In parallel with critical limits, laboratory scientists must monitor analytical linearity, which defines the dynamic operational range where electronic detector output remains directly proportional to cell concentration. Operational linearity will depend on the specific analyzer. Examples of linearity include values spanning 1.0 to 99.9 times 10 to the third per microliter for WBC, 0.30 to 7.00 times 10 to the sixth per microliter for RBC, 0.1 to 25.0 grams per deciliter for hemoglobin, 10.0 to 60.0% for hematocrit, and 10 to 999 times 10 to the third per microliter for platelets.
If a parameter falls below operational linearity, electrical impedance background noise can interfere with true cell detection, requiring a manual hemocytometer chamber count or a manual spun microhematocrit for accurate reporting. Conversely, when cell counts exceed linearity, the analyzer displays an overrange flag of ++++, signaling that an automated dilution protocol must be executed.
Let us review the mathematical formulas used to derive red cell indices from primary analytical measurements. Mean cell volume, or MCV, expresses the average size of circulating erythrocytes in femtoliters. On analyzers utilizing electrical impedance, MCV will be derived from the RBC histogram. Otherwise, MCV can be calculated by multiplying the hematocrit percentage by 10 and dividing by the RBC count.
Mean cell hemoglobin, or MCH, reflects the average weight of hemoglobin inside an individual erythrocyte. It is calculated by multiplying the hemoglobin concentration in grams per deciliter by 10 and dividing that product by the RBC count.
Mean cell hemoglobin concentration, or MCHC, measures the average concentration of hemoglobin within a given volume of packed red blood cells, expressed in grams per deciliter. It is computed by multiplying the hemoglobin concentration in grams per deciliter by 100 and dividing by the hematocrit percentage. Alternatively, MCHC can be derived by dividing MCH by MCV and multiplying by 100.
Lastly, the RDW-CV expresses the degree of red blood cell volume heterogeneity, or anisocytosis. It is mathematically calculated by dividing the standard deviation of red blood cell volume by the MCV and multiplying that quotient by 100 to obtain a percentage.
Let's walk through a concrete numerical calculation using a standard patient profile where the RBC count is 4.50 times 10 to the sixth per microliter, hemoglobin is 13.5 grams per deciliter, and the hematocrit is 40.5%.
To solve for MCV, we multiply the hematocrit of 40.5 by 10 to get 405, and then divide by 4.50. This yields exactly 90.0 femtoliters, which sits squarely within the normocytic reference interval of 80 to 100 femtoliters.
For MCH, we take the hemoglobin value of 13.5 grams per deciliter, multiply by 10 to obtain 135, and divide by the RBC count of 4.50. This calculates to 30.0 picograms, placing the cells in the normochromic range of 28 to 34 picograms.
Finally, to calculate MCHC, we multiply the hemoglobin of 13.5 by 100 to get 1,350 and divide by the hematocrit of 40.5. This yields 33.3 grams per deciliter, which confirms a normochromic erythrocyte population within the 32 to 36 grams per deciliter interval.
Moving to leukocyte differential mathematics, clinical evaluation requires converting between relative percentages and absolute counts. The absolute count in thousands per microliter is determined by multiplying the relative percentage divided by 100 by the total WBC count.
In our first worked example, a patient has a total WBC count of 11.9 times 10 to the third per microliter and a relative lymphocyte differential of 23%. Multiplying 0.23 by 11.9 yields an absolute lymphocyte count of 2.7 times 10 to the third per microliter.
In our second worked example, we perform the reverse calculation to determine relative percentage from an absolute concentration. Here, the patient exhibits a total WBC count of 25.1 times 10 to the third per microliter with an absolute neutrophil count of 9.3 times 10 to the third per microliter.
By dividing 9.3 by 25.1 and multiplying by 100, we calculate a relative neutrophil percentage of 37.1%. Tracking absolute concentrations is essential because relative percentages can appear spuriously altered during marked leukocytosis or leukopenia.
While reviewing CBC results from our automated analyzer, we utilize the rule of threes as an internal quality control check for sample results. Under normal physiological conditions, the RBC count multiplied by 3 should approximate the hemoglobin concentration within a plus or minus 3 margin.
Furthermore, multiplying the hemoglobin by 3 should approximate the hematocrit percentage within a plus or minus 3 margin. If a patient's results fail this mathematical relationship, or if the calculated MCHC exceeds 36.0 grams per deciliter, the results must be held and investigated further.
Discrepancies in the rule of threes indicate either red blood cell pathology, such as hereditary spherocytosis, or analytical interferences like cold agglutinins, lipemia, or unlysed erythrocytes. The laboratory scientist must investigate and resolve the discrepancy before releasing patient results.
The classification matrix organizes red cell morphology into distinct diagnostic quadrants based on cell volume from MCV and hemoglobinization from MCHC. Cells with an MCV under 80 femtoliters are microcytic and appear smaller than a resting small lymphocyte nucleus, while cells with an MCV over 100 femtoliters are macrocytic.
Regarding hemoglobin concentration, an MCHC below 32 grams per deciliter defines hypochromia, where the central zone of pallor exceeds 1/3 of the cell diameter. An MCHC between 32 and 36 grams per deciliter is indicated as normochromia, with a normal central pallor.
An MCHC exceeding 36 grams per deciliter is flagged as hyperchromic, but true physiological hyperchromia cannot occur because hemoglobin precipitates at higher concentrations. High MCHC indicates either genuine spherocytes with decreased surface area to volume ratio or analytical interference.
Microscopic review of a well-stained peripheral blood smear provides essential quality control to validate automated cell counts. All microscopic estimations must be performed exclusively in the monolayer, where red cells are evenly spaced and touching without overlapping or wide empty gaps.
For the leukocyte estimate, we count WBCs across 10 consecutive fields using the 40x high-dry objective. Summing these counts and dividing by 10 gives the average WBC per field, which is multiplied by 2,000 to yield the estimated count in cells per microliter.
For the platelet estimate, we examine 10 consecutive fields under the 100x oil immersion objective in the monolayer. The average number of platelets per field is multiplied by 20,000 to calculate the estimated platelet count per microliter.
These visual estimates must correlate with the automated counts before results are verified, providing a rapid check against electronic counting errors, undetected clots, or extreme cell aggregation.
When analytical interferences occur, they produce distinct CBC abnormality patterns that point directly to the underlying mechanism. In lipemia or extreme icterus, turbidity in the optical measuring chamber increases absorbance at 525 nanometers, causing falsely elevated hemoglobin, MCH, and MCHC, while RBC count and hematocrit remain accurate.
When cooled agglutinins involved,
IgM autoantibodies that cause red blood cells to clump at room temperature, the analyzer counts each clump as a single large cell, resulting in a falsely low RBC count, a falsely high MCV, and an elevated MCHC above 38 grams per deciliter, while total hemoglobin remains accurate.
Gross in vitro hemolysis involves pre-analytical lysis of red cells in the collection tube. The analyzer records a falsely low RBC count and calculated hematocrit, leading to spuriously elevated MCH and MCHC, even though the hemoglobin reading reflects true total released hemoglobin.
Platelet clumping and EDTA-induced satellitism cause platelets to aggregate or adhere to neutrophils. This drops the measured platelet count while falsely elevating the WBC count as large platelet clumps exceed the leukocyte sizing threshold of 35 femtoliters and enter the white blood cell counting bath.
To resolve optical interference from lipemia or severe icterus, we perform the plasma replacement protocol. The well-mixed whole blood sample is placed into a Wintrobe tube and centrifuged for 5 minutes at 2,000 revolutions per minute to pack the red cells.
The operator marks the exact upper plasma meniscus on the tube wall, then carefully aspirates and discards the turbid plasma layer without disturbing the buffy coat or erythrocyte column. Isotonic analyzer diluent is added back to match the original meniscus mark.
The tube is gently mixed by inversion to resuspend the cells and reanalyzed on the automated instrument. The laboratory reports the corrected RBC count, hemoglobin, hematocrit, and red cell indices from the replacement run, while reporting the original WBC count and platelet counts from the initial unmanipulated run. When a patient's cell count exceeds the instrument's operational linearity, the analyzer displays the +++++ flag. The laboratory scientist must execute a manual dilution protocol using isotonic diluent.
The first step is to recognize the overrange flag. In step 2, the laboratory scientist pipettes 500 microliters of well-mixed whole blood and mixes it with 500 microliters of diluent to prepare a precise 1:2 dilution.
In step 3, the diluted sample is reanalyzed, and all directly measured diluted parameters, including WBC, RBC, hemoglobin, and platelets, are multiplied by the dilution factor of 2. If extreme leukocytosis is present, the RBC count must be mathematically corrected by subtracting the WBC contribution, and a spun microhematocrit should be performed on original undiluted blood to recalculate indices accurately. Let's illustrate another important instrument-related error, the failure mode caused by an exhausted or unmetered lytic reagent supply. When the lyse reagent fails to dispense into the measuring baths, unlysed erythrocytes remain intact throughout analysis.
In the leukocyte aperture bath, millions of intact red cells enter the sensing zone and register across the lower WBC thresholds. This generates a spurious leukocytosis, often exceeding the instrument's linearity, and a false lymphocytosis flag.
Simultaneously, in the spectrophotometric chamber, intact unlysed erythrocytes scatter light and create severe turbidity, driving absorbance to maximum levels. This produces a falsely skyrocketing hemoglobin value above 25 grams per deciliter, leading to catastrophic rule of threes failure and an impossible MCHC exceeding 50 grams per deciliter.
Maintaining analytical integrity requires strict adherence to daily quality control, preventative maintenance, and biosafety standards. Each day begins with running particle-free background counts, verifying the WBC background is 0.1 or less, RBC is 0.02 or less, hemoglobin is 0.1 or less, and platelet background is less than 10.
Multi-level commercial controls spanning low, normal, and high concentrations must be evaluated using Westgard rules, holding patient testing if values exceed 2 standard deviations. Scheduled maintenance, including weekly deproteinization and monthly aperture flushes, prevents protein buildup and aperture clogs.
Pre-analytically, EDTA specimens must be mixed for 2 minutes and inspected for microclots using applicator sticks, rejecting clotted specimens immediately. Finally, all laboratory personnel must follow universal precautions, wearing gloves, lab coats, and splash protection when handling biological specimens and hazardous waste.
Today, we reviewed automated CBC parameters, mathematical derivations for RBC and WBC indices, critical limits, linearity, troubleshooting, and quality control protocols.
Thank you for joining us.